According to BCC research, the global microbial natural products market is going to accrue up to $346.6 billion in 2027. If you look at it, it's a huge amount of money. But I'm here to tell you that we stand a chance of losing out on getting up this milestone. And that's why I chose the topic to share my perspective on what I titled The Economic Trade-off of Studying Microbes as Simpletons. Why exploring the complexity of their crosstalk is paramount for innovative discoveries. If I walk down the street and talk to someone, anyone at all, and ask them what they think about microbes, I'm sure that what image that would come to me or could be described to me will be something similar to that. Microbes are monstrous, they cause diseases. But most importantly, they cause diseases upon contact with anyone that they come in contact with. This is a general saying, and I don't blame anyone for that, because the media has always projected that. But I'm here to tell you that microbes are much more than that. Microbes are highly beneficial to us. In fact, they influence every aspect of our economy. They help us to grow crops. They help to maintain our food, enhance them, and preserve them. Think about your yogurts, your sauerkrauts, and everything that you eat at home that are kind of connected to microbes. Microbes, and we are beginning to get information right now that gut microbiome kind of shaping our overall well-being. Microbes also help our environment. They degrade toxicants in the environment. Microbes have revolutionized our industrialization and has also influenced our policies. If you think about it, COVID has done a lot more harm than good to our policies, right? But then microbes are very social organisms, and they are never ever alone. In fact, the entire Earth is covered by microbes, but they appear in stratified and yet functional micro-communities where they interact with friends, families, and foes, as depicted in the picture, in the image that I have here. This social construct is well described by Davy and O'Toole, 2000, where they show that microbes are usually clustered on the basis of their nutritional needs, as well as their ecological function. Take, for example, the simplest form of energy that microbes usually assimilate from the environment are in the forms of simple sugars. But not all microbes in the environment can do this. And so oftentimes you find that microbes are capable of interacting with the ones that are unable to degrade our ferment sugars, are usually found in close proximity with the ones that are capable of degrading sugars, so that they can feed off of their waste, which are organic acid in this case. More so is that the ones that are incapable of also utilizing organic acid can feed off of the remnants from organic acid degradation, which in this case could be hydrogen or other basic elements like carbon that constitute organic compounds. But nutrition is not the only reason why microbes form this micro consortia. Microbes are able to communicate with one another using signaling compounds. And these compounds can cut across members of the same group or even across members of distinct groups. On our bodies, we are made up of much more than 1 trillion bacteria that are found on the surface of our skin and even in our embodied orifices, such as the mouth, our stomach, the intestine, the urinary tract, and also our skin. These orifices are laden with microbes, such that when any pathogen comes in contact with these orifices, which form the basic mode of entrance or route of entrance for infections, microbes are able to battle with them. And if any pathogen is able to overcome these microbes, resident microbes in these orifices, then an infection can occur. But I can also assure you that if you take a swab from your skin and give it to someone to look at it in the microscope, or even place it out in the laboratory, you're going to find a collection of multiple microbes on the plate. But why then do scientists pick out these microbes as individual colonies and study them, grow them, and then take them for experimentation, when we all know that these microbes are never alone? And so taking one single colony does not really represent what we have in that consortia. And it doesn't also give us the benefit of truly understanding the intricacies of their existence in that community. And so I have looked through literature, and I'm beginning to see that the study of microbial consortia or studying microbes in their communities is gaining traction. And there are very important approaches that could help us to gain more insights. And one of those approach that appeals to me is what I would call the top-down approach, where you could collect a collection or a community of bacteria and begin to observe them based on what changes that occur or what metabolic implications of that community could be, and then begin to trickle it all the way down to identify who are the key players within the community. On the other hand, you can also flip this approach and look at them one after the other, building up on that collection until you get a community and gain much insight into what every individual could be doing within that community. That way, I can assure you that this method has been adopted, and it led to the discovery of a novel natural product, an antibiotic that can destroy Staphylococcus aureus. But that's not the only importance of this observation. But what was very critical in this research is that they found out that 2 microorganisms rather than 1 are capable of uniting together to make this happen. Otherwise, when they're separated, this can never happen. And so that's the implication of studying microbes in their communities. We gain insight into more things, novel things that were never existing before. Paying more attention into the ecological construct of microbes have also taught us that plants are able to communicate with their resident microbes in the soil, right very close to their rhizosphere. These plants are able to send out signals to them, to these microbes, calling for help when they need it, especially under stress conditions. And so taking into consideration all of what I've told you, you will agree with me that singling out microbes and studying them in isolation would take time and consume more resources, but at the same time, we strip them of what makes them who they are within that community, and we never get the opportunity to gain insight into what their function within that community could be. So I am of the proposition that we could adapt or create a more complex but systematic approach that can allow us to study microbes in communities so that we can gain novelties or make discoveries that can help us boost our economy. Thank you for listening.